Hi, and welcome to this very special Impetus interview. Today we are joined by Joan Casals, who is an agriculture engineer with a PhD in agri-food engineering and biotechnology, and Tony Granell, a research professor at the National Research Council in Spain, who focuses on plant molecular genetics in tomato fruits. And they're going to be talking to us today about the HARNESSTOM project. Thank you very much for joining us today.
Thank you.
Thank you
for having us.
So just as a quick introduction to the project, could we talk about the work that HARNESSTOM has done to help protect food security and ensure, and the work that you've done breeding tomatoes with and working with citizen science, I suppose.
Okay, yeah. In the last already 3 years, we have been focusing our attention to develop pre-breeding materials so that tomatoes that we eventually will produce will be able to cope with the stresses associated with climate change, for instance, high temperature or drought. And also, there are emerging diseases that are affecting tomato production worldwide. So we are trying to develop varieties that introduce from wild relatives disease-resistant genes that will make these tomato plants able to fight these new diseases. Also, we are concerned about the fruit quality because consumers are concerned about fruit quality. So we are also focusing in improving the flavor and nutritional composition of our tomatoes. And lastly, there is a lot of activities going on citizen science, and Joan will go in more detail to these aspects. So how citizens think about tomatoes and how we can introduce in our breeding how they feel about tomatoes.
That's really interesting. So you've been working with these communities to breed more hearty, more flavorsome, more nutritional tomato species, and working with the farmers to, allow them to gain access to this genetic sort of data that allows them to breed other tomato varieties, I suppose. But I suppose one question is, this might seem a bit of a obvious kind of question to you, but why tomatoes exactly? Why not another plant species?
Well, why tomatoes? Because there's a lot of information, a lot of knowledge, and also a lot of skills in the field. There are a lot of breeding companies, and tomato have been used also for, in many cases, as a model species. So when there are problems, for instance, drought or increased temperature, you can use. Of course, you're interested in your own crop, but if you still have to do a lot of studies to understand what are the basis of why the tomatoes don't grow, why the plant doesn't grow well in particular stress condition, you better use a crop or a plant that is the best suited. And tomato is the best suited in terms of genetic resources, diversity, skills, and methods, and genetic maps. We have a huge diversity. The seeds of this diversity are stored in seed banks. These are a community of scientists in Europe and elsewhere that have been working in tomato for a long time. So we have a very basic knowledge that maybe you don't find in other crops. Also, tomato is a very important crop. As the most, let's say, the horticultural crop probably provides most of our healthy compounds that we need in our diet. So it has a number of, let's say, components that make it so very useful for scientists to address very basic questions, and it's also very applied questions.
I suppose I used to work at an agricultural university, so I was quite surprised to find how much brassicas, like all species, like a lot of farm and agricultural species of plants and the foods we eat come from brassicas, but it seems very similar to tomatoes, I suppose. They're related to the nightshades, deadly nightshade, but like tomatoes and potatoes and chilies, they all come from a very similar kind of family.
Of course, yes. Tomato was the first Solanaceae crop to be sequenced, and the idea of developing a high quality genomic sequence of tomato that could be used. This knowledge could be used for many other Solanaceae where there's a lot of genetic synthesis. So there is a lot of things we can learn from tomato that can be applied to other solanaceae crop and eventually to other crops. So for instance, tomato is the model species for climateric fruit ripening species. Like every fruit that ripens very fast after you pick it from the plant, tomato is the model, and of course it has to do with the storability of the product, with the quality of the product after you pick it up from the plant. So basically, you can have, there are many models plants for different type of crops. Of course, tomato cannot be a model plant for a cereal. You have to work on a cereal, cereals are important. But if you have to work for a vegetative crop, that horticultural crop that is providing us with a lot of the nutritional component that will make us healthy, we work with tomato as a choice, as a best choice.
Mm-hmm. So I suppose just due to the fact that we have selectively bred these plants from their wild varieties to the things that we see today, we've lost quite a lot of the genetic makeup of these wild types, I suppose. And as kind of our world keeps changing at a rapid rate nowadays, and our requirements for supplying food change, this loss of genetic diversity has impacted kind of the variety of kind of characteristics that we can get out of the fruit. I'm just wondering if you could talk a little bit about the impact of this selective breeding and kind of the idea of the ideal tomato that we now see, but yeah, the loss of other kind of key genetic kind of components, I suppose.
Yeah, when we look at this project and other projects, European projects that have been funded by the European funding agencies, we look at the genetic diversity, and it's clear that when you compare the genetic diversity in Solanaceae and Solanum lycopersicum, which is the tomato as we know it, and you compare it with the wild relatives, there is much less diversity. Also, if you compare the diversity in Europe versus the genetic diversity in the closest wild relative to tomato, that is Solanum pimpinellifolium, there has been a lot of loss of diversity. And the scientists believe that there have been like two bottlenecks in which diversity has been lost. But it's also true that during in Europe, that is considered the second place for diversification of tomato, apart from the Mesoamerica. It has been also an increase in diversity, probably because of this. During these 400 or 500 years, farmers have been selecting those tomatoes that are adapted better to their conditions. So there has been a small increase in diversity. Also, in the last 50 years, breeders have introduced diversity by crossing with wild relatives, trying to increase, of course, this diversity to produce plants that adapted better to new conditions or suited better the consumer's needs. So let's say that as any other crop, always when you do a domestication from wild relatives, you lose diversity, but there is also very targeted increase in diversity for traits that farmers or consumers or humans are interested in. So in general, loss of diversity, but when this has the highest impact when it comes with diseases, when all we new challenges like this huge increase in temperature in the summers or extended or cold temperatures in winter associated with climate, then is when we have to go back into the wild relatives to find whether during the evolution the wild relative have already developed let's say the genetics necessary to deal with these issues.
That's really interesting. Could we talk a little bit about how you've been working with the farmers community, the farming community as a whole, and how you've been working to explore these varieties of tomatoes, I suppose, and how they've been able to access this information?
Yes, Chris. So an important objective of HARNESSTOM is to transfer the results, but also the materials that are being developed in the project to farmers and also to citizens. As Tony mentioned, in the HARNESSTOM project, there is one more package that is dedicated to citizen science, but also, let's say, farmer science. This the approach, the name is participatory plan breeding. So in this work package, we're using kind of a circular approach. First, we asked citizens how they like tomatoes. So we wanted to gather information about their preferences regarding fruit color, sensory aspects, or fruit shape characteristics. Once we had this information, we went to databases that were created in previous projects, and we filtered the databases to select some materials that they can fit on these consumer preferences. And then we started to work with farmers. And with farmers, we proposed to them, we gave to them plantlets of these varieties that were selected, and we worked with them to study the adaptation of these varieties to their fields. Once we had then let's say selected these promising materials, we came back to consumers, and then we offered to consumers fruits of these varieties, and we asked them about their preferences. So this is kind of a circular work where we start asking consumers what they like, then we work with farmers to select varieties, and then we come back to consumers to offer them these varieties. And this has been the main objective of this work package. And we can say that we have been collaborating with around 5,000 persons in this approach. And the works have been done mainly in Spain, but also in Italy and Bulgaria.
So it feels like there's this real active feedback loop within the communities and the farmers, I suppose, because obviously the farmers might want more pest-resistant varieties and that sort of thing. But I suppose the ultimate goal is to have a tomato that tastes good and people want, I suppose. So you can breed in kind of these other factors, but one of the overarching kind of features of the tomato is it has to taste good, I suppose.
Yeah, this can be also a second step. I should say also that we are working also with farmers to update the characteristics of some old run races that they are interested on. And with these specific case studies, we are doing plant breeding programs to introduce these resistant genes to these old varieties, and to, let's say, to promote the recovery of the cultivation of these old land races. But this is more an approach that we are using with specific local farmers that they're interested in these old land races, and they want to target some consumers, so to promote also the local agriculture.
So after this process of like this feedback within the communities and the farmers to develop kind of these varieties, I suppose, what happens to the data afterwards? Is like, are the seeds for these varieties provided to farmers? Do they have this database of, yeah, how does the farming community use this data after the project, I suppose?
You go with the farmers, and I will go with the general about the database we are producing. Okay,
Maybe
John?
start with the general database, Tony.
We are building what we want to be the most comprehensive database for tomato, for the genetics and phenotypes of tomatoes. So that and this will be of interest for a huge diversity also of people, from breeders to farmers to scientists to people in general. And this will include more than 30,000 different accessions of tomatoes and characteristics for over more than 500 traits, including metabolites and fruit shape, fruit size, productivity. So traits that are interesting for the different stakeholders. And this database will be open to everybody. It's already open for some aspects. It's called HARNESSTOM database, and it will be open to everybody after the finishing of the project. And for the developing of the materials and the access of these materials to the user, to the end users, whatever people are interested in this, there are two types of materials we're developing. One is the materials that we are developing in close collaboration with breeding companies, in which the knowledge that we have in the academia groups, or in research groups, like knowledge about where the genetic basis for increased resistance to these diseases lays, like in this wild relative. And then we are working together with the breeding companies to produce varieties that have increased resistance to the new disease or to improve flavor. And when we use the elite material from the breeding company, of course, this material eventually will be developed to end up with a new variety, and this will be done by the breeding company. So the access of these materials is restricted to the seed company that eventually will produce a product in the market. Another issue is the knowledge and the materials that are developed in the frame of close collaboration with farmers. And this deals with another type of tomatoes, okay? And this also relates to the discussion that John was introducing before. We can go with, people complain about tomatoes and tomato because they lost flavor. And there's also very traditional variety they highly appreciated because they have a high quality or very good flavor according to the local people that normally consumes these tomatoes. So we can go two ways. Either we try to get flavor back into the modern varieties that have been basically improved in terms of disease resistance to new diseases. And this has been excellent work done by the company, by this breeding company. Also, they are highly productive. They produce throughout the year. They respond very well to the inputs of fertilization. And this is one approach. We use those elite materials. This is what we are working with the breeding companies to improve flavor or to improve all the characteristics. Or as the approach that Joan is doing, we go from the traditional varieties, trying to introduce disease resistance while maintaining fruit quality. But I will leave then Joan to elaborate a little bit more how they are working on the farmers and the citizens to make knowledge and the materials accessible in the future or now already in the interaction with the farmers.
So with regard to materials that they have been developed using these participatory approaches, for sure we need to guarantee and to promote the open access condition of these materials. And for this, we're working close with these networks that we have developed in this work package about citizen science and participatory plant breeding. And we are already transferring the seeds and the plant materials to these farmers with this huge network that we have created in Spain, Italy, and Bulgaria. But we are also contacting some organic seed cooperatives to promote that these varieties, that they can go through commercialization and to reach a higher number of farmers. Finally, with these specific case studies, these local land races that we are working to introduce these resistant genes. For these cases, we're working with local cooperatives that they will provide for the work that we have been done breeding these varieties. So we are using a multiple approach, to transfer and to guarantee the open access of these materials.
Mm-hmm. No, that's really interesting. I'd want to just kind of go back a step, I suppose, just slightly. You talk about the selective breeding and the creation of these mixing of tomato varieties. Is this done traditionally through like just crossbreeding with like as you'd see in the wild, or are you using genetic editing techniques to splice these genomes together?
We have like three big breeding programs. One trying to address the issue of climate change, the other with emerging diseases, the other fruit quality. In all those cases, we're starting from the knowledge from the academia, where the QTL, with the region of the genome that can be used to increase, for instance, resistance to the disease, or to increase tolerance to that stress, or to have better flavor, or to increase this compound that give better flavor. And this is a traditional approach by back crossing and selecting materials using genetic markers. Okay, so it's crossing and selection guided by genetic markers that we know are linked to the trait. So cross and selection and guided by markers. What generally is called marker-assisted selection breeding. Although we still do work on editing, genome editing for particular cases, And we are strong supporters of these techniques, because not only because it speed up breeding, it's sometimes, and this is something we realize, for instance, when we screen for resistance to emerging diseases, or we try to find genes that increase tolerance, the amount of genetic material that we can screen is very limited. When you go and it's not like it's not going to end. There is a limited amount of accessions that are in the gene banks or the seed banks, like in TGRC in UC Davis, or the COMAF here in Valencia, or in AVRDC in Taiwan. And these materials, they were collected from the wild, from Mesoamerica, or from elsewhere, everywhere in Europe, in the case of tomato. But if you want to go to the wild relatives, you have to go to Mesoamerica, South America, Central America. And this is very limited amount. Most of the wild relatives that were collected in the 50s by Rick in California, they are not there anymore in the wild. So the only seeds that remain from those wild accessions are now at the seed banks. And we go there and we get the seeds, we screen them for to find any resistance. We don't find any source, or maybe one or two. Okay, so the amount of diversity is limited, and it's limited because the efforts to get these wild relatives into seed banks have been limited. And of course, there is the limitation, natural limitation, because there has the place. Imagine that most of the material that are still available are in deserts, or places where there is no population increase or no farming activities, and this is why it's still there, the wild relative. Okay, so farmers and wild relatives are, let's say, always in fight with each other, because they need them, but they also want to use the land. Farmers want to use the land. So if you use the land, you don't have wild relatives. But the wild relatives are now there, are in there. It used to be Central and South America, and most of the germoplasm that is available for breeders are those limited amount that are in the seed banks. And you realize that how limited this is, because despite we scientists say, oh, there's a huge diversity, and it's true, this limitation means that sometimes you try to find diversity that is useful for you, and is not there, because mainly during evolution, the wild relative never face similar situation. This is a new pathogen. Maybe there is no disease resistance because the wild relatives never were exposed to these pathogens and didn't develop the appropriate resistant genes. So we need to introduce diversity. And one way to do this is through knowledge. Once we know more, we can then go and edit it and edit the gene that may increase resistance. And in the case of disease resistances through genome editing, the approach is to knock down susceptibility genes. And it's another complementary approach of the so-called disease resistance to, let's say, eliminate one or several of the susceptibility genes that eventually result in a plant that is more resistant, although maybe still replicate the pathogen. So genome editing is also one of the approaches we are using to increase the speed of breeding, because your ability to develop a variety that is better than the one before, and is able, for instance, to fight new stresses or new climate change conditions that compromise your cultivation, is a matter of time. You need to go from one cross to another. You have to produce a seed. This seed, you plant the seed, you get a plant, you get a flower, you cross this flower, this flower with the pollen from another flower, you get this, and you select using marker. But you still need to do crosses, and you need to go from seed to seed. And genome editing is proving also a useful technique to edit the plants and develop a variety that it goes from seed to seed faster than a normal variety. So you can go through the 6 or 7 back crosses in a much faster way. In addition to, you can go also by editing, that you don't need to do so many crosses.
I suppose that just goes to show the importance of these gene banks and these seed banks in conserving kind of these wild varieties. And as as we all know, we're we're experiencing an extinction event and loss of habitats. And yeah, having access to these old varieties and this genetic database, I suppose, is going to be a key going into the future, I suppose.
Yeah, I think it goes hand with hand. I mean, having your money in a vault in a bank and not making it to work is useless because it's there. You have to produce to do work with them and to put them in the market. And the same with the seeds. It's good to have a storage of seeds, but it's good as much as it's used by breeders so that they eventually end up with the new varieties. But in order to do this, the best is to have data associated with the accession, with the each of the seed batch you have in the storage room. Otherwise, it's a number, and you don't know what you have there. That's why we are making a huge effort into developing this database that links the seeds in the data in the seed bank with the phenotypes, with the genetic composition of the seeds of each of the accession, and eventually will lead the new generation of breeders in a much easier path. And also, I think Joan can tell you about the work they have been doing in this respect.
Yeah, I think also an important issue is that all these thousands of varieties that they are stored in germplasm banks, we need to develop strategies to allow farmers and citizens to fish, to be able to have to access these materials, and this is not easy. And this, I think, it was one of the objectives of the HARNESSTOM project, is to try different strategies to connect farmers with seeds and to allow them to select some of the materials and try to produce them and try to sell them to consumers. So this was what we have been dealing the last years, to try different strategies just to identify the most effective ways of transferring and recovering the use of these materials that are in germplasm.
I suppose going back to the work that you're doing with the consumers and the farmers as well, the stigma around genetically engineered foods and that sort of thing has reduced over the last few decades as it's become more and more integrated into the research and science that we do. I'm just wondering if there's still were there any kind of, I don't know, issues with the consumers when you brought up the fact? Is there still kind of an underlying kind of uncertainty around genetically modified food, even though it's just a new version of crossbreeding that we've been doing for hundreds, thousands of years?
Really, I cannot answer your question because, as Tony mentioned, in the participatory plant breeding programs that we have developed, we have used this marker-assisted selection. So this means that we have been using, let's say, the classical crossing strategy to obtain new varieties. And these are the varieties that we have developed and that consumers have evaluated and farmers have grown in their orchards. So we didn't obtain a response to these questions. But maybe, Tony, in other work package, yes, that we have been studying this approach.
In the dissemination work package, in which we have been very active, particularly in the case of genome editing in tomatoes, in plants in general, and also to bring to communicate to a large audience what are the benefits and the importance of the new genomic techniques, including our perspective that genetic diversity is limited and we need to produce more. And also we need the breeding is producing a timeline that we need maybe to shorten. And so we need to either to introduce technology that do not require so many back crosses and go directly to the trait. And this can be genome editing, also, or introduce genomic editing to shorten the cycle from seed to seed. So in many cases, and we have been very active addressing from scientists to breeders to consumers to public at large, but also farmers. In a particular session, we have to let them know what is the new genomic techniques and the situation in Europe and the new legislation that is now under evaluation at the European Parliament and in Europe. So, and And the feedback that I got from many farmers association, even individual farmers, is they would like to get access to these technologies, not so much for the technology, but because what they're interested, they are not concerned about the technology. What they are concerned is to produce a product they can put in the market with the same in the same conditions that the competition from third countries. And Europe cannot be an island for the new innovations by genetic engineering or by the genome editing, as it is the current situation, because at the end, are the farmers losing competitiveness in face of the competitors in other places. So the feedback was not much of a concern because of the genetic engineering, let's say, contaminating their stocks and their abilities to sell. And of course, we're also working with organic farmers that they're let's say, not able now to use genome edited plant, but some of them, they would be interested because some of the edited plants maybe are better for not using pesticides because they have their own way to fight bugs or pathogens. So, but the other farmers, they are not in this sector of production. They are also willing to have whatever they provide them with a more competitive product. And so all the approaches that we are using in this pre-breeding programs and on tomato, that we are using, there's a classical marker-assisted selection using wild relatives. In some cases, once we get down to the gene, then we can go very fast by genome editing and shorten the time of breeding. And the farmers for sure are waiting for the EU to let's say, to waive and to say we are happy to have this new piece of legislation that will enable scientists and farmers and breeders to put in the market in the people's houses better tomatoes or by large any vegetable improved by these techniques.
So the HARNESSTOM project was received a honourable mention at the 2023 EU Prize for Citizen Science. I was just wondering if I could ask if this honourable mention impacted the work that you're doing and what the experience was like to be included in this process.
We are very happy to receive this honorary mention. And I think the most important thing that we obtained with this honorary mention is to know about the other projects. This permitted us to see what other colleagues were doing and which methodologies they were using to implement citizen science, even in scientific aspects that are really far from our tomatoes that we are dealing in harvest. So this, I think, was an important thing, to know other citizen science projects that are being doing in Europe. Apart from that, for sure, the visibility that gave us this honorary mention is very important. And we have been also helping with other projects to start some other collaborations. And also, I think the conversation that we are having today is a good example of what we are obtaining through this mention in these citizen science awards.
And I suppose
one important thing, Christopher, is that this prize also recognizes that scientific projects like ours are not only interested in the traits that are of the interest of breeding companies that are interested to sell seeds and they are done with the job. Eventually, what we are trying to say is that consumers matter, farmers matter, everybody matter. So it's important that we take in consideration, and this work package is the one that has the highest impact in terms of people who show interest for our project, because this is closer to the people's interest. Whether we in the back of, whether we do also this targeted project with the breeders is okay. But the breeders, of course, they want to produce a variety that is, when you put them in the markets, is purchased and it has an impact in the society. But of course, there's always this idea that what happened eventually with the loss of flavor or with the loss of this traditional flavor is because breeders have followed the easy way of breeding for product, for yield or for the traits that are of interest of breeders, of seed companies and farmers. But we in this work package, we show that we want to take in consideration, and also the information that we get in this work package is highly relevant for the breeders too.
So yeah, that's really interesting. Yeah, and I just want to ask my final question, I suppose, is what comes next for the HARNESSTOM project? What short term and the long term? Yeah, what's next on the horizon?
Well, it's not that when you finish a project, everything is finished. I mean, breeding is an ongoing project, an ongoing thing that will end when we finish as a as a as a society. It's breeding is associated with agriculture. With agriculture is responsible for what we are now. Otherwise, we'll still be spending 90% of our time gathering for fruits and nuts in the wild. So the idea is that we have to continue our efforts to eventually give the consumers what they need, the farmers what they want, and everybody be happy with what we put them in the market. So the products that we produce are the healthiest possible, they make our lives better, and more. And we are happy because the taste of the 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 tomatoes are the best we can provide. And this, despite the circumstances that, for instance, productivity costs have increased a lot
Hmm.
because of everything increases in price. And we want people to consume tomatoes because they're a healthy product to consume, and this gets you a healthy life. And if the price of the commodity increases, people may start to stop buying buying vegetables and fruits, and then and we need to give a healthy life and consume good and healthy products. So again, this means that we have to improve tomatoes, and by uh, uh, and this is an ongoing process, and we will face new challenges, and it's not that we are done at the end of the project. The idea that we will provide seed companies with improved materials for some of these traits, and farmers with improved materials for resistance to emerging diseases, and everybody will be happy. But this is an ongoing project, an ongoing thing. We are not done after the project. And of course, the database, for instance, we are trying to have a database that contains all the information available for tomatoes, uh, uh, and be open to everybody. Uh, this database cannot be say, okay, everything that we need to know is there. No, it is. We have to populate this database with new data. This means new project, new work, new people involved, and enthusiastic about providing this information to everybody.
With regards to the results that we have obtained using these participatory approaches, I think also we need to maintain these efforts to transfer the materials to farmers, citizens, to reach the consumers. I think we need to maintain the communication, so to explain the results of the projects, both to the scientific community, but also to the general audience. And I think also we should start to work and to transfer the methodologies that we have been working on to other crop species. Why not to also to collaborate with researchers that they work with other species and use and to share with them these citizen science and participatory methodologies that we have been working in the HARNESSTOM project.
No, that sounds very exciting. And yeah, that's my last question, I suppose. So I just wanted to double check. Is there anything else you'd want to mention just before we close this conversation? No?
I think, as you know, with the current situation about the wars and everything, food safety and food security both are more important than ever. So I think Europe should be able to develop varieties by themselves, not rely on other people. Don't rely on other people, the technologies. We have to be able to develop varieties that satisfy all the Europeans' needs without having to rely on the supply of food by third countries. And this requires continuous innovation, because the challenges change and because the needs of the people change.
And I think that's a very interesting way to end this interview. So I just want to say thank you very much for joining us both.
Thank you, Chris.
Thank you, Christoph.
And yeah, we'll speak to you soon. Bye for now.